Okay. What is this? Start AI companion?
Say no.
No, we don't need this. They got more bells and whistles now. Anyway,
Some of it's helpful, but
yeah. I'm gonna just do a brief review of what we did last time.
so welcome, Eric.
Yeah.
Thank
The
you.
rather abrupt introduction.
Nice to see you
Oh, I
guys.
see what happened. Marjorie
I don't
took
know
me.
how
Hold
long
on.
I'll be able to stay on, but I'm going to stay on as long as I can.
No, I understand.
I
Let me just go
finished a
to
12-hour work
Oh,
shift.
I'm just gonna show one thing and then we're gonna go off into the class. So here, okay, here we go.
We're happy to see you.
Thank you.
Okay.
Feelings mutual.
So this is a new telescope that just came online. It's down in Chile, and it's named after Vera Rubin, who was one of the people that dealt with what is called dark matter. And she did a lot of astronomical observations that showed that, yes, there's a lot more dark matter than matter. And this telescope that's here just got cranked up. And here's a picture here of its first photo. So it took a look out into space. It's got 3.2 billion pixels in it, which is, it's basically the size of a small SUV. The pixels. Okay, this is amazing engineering to do this.
And what can that telescope do that Hubble or Webb cannot?
Although those are in orbit.
Right.
There's the James Webb
ground-based.
telescope.
I understand.
This is ground-based, yeah. So this is high enough in the Andes so it can get a good shot at all this stuff.
Yeah.
But anyway, the only reason I put it in here was that there's a former colleague of mine from Bell Labs, Tony Tyson, who's the guy who actually honchoed this thing. He's now at the University of California at San Diego, and he started working on this in the mid '70s. Okay, and he's been working on it ever since. So like they charge a couple devices you have in your phone for your camera in your phone came out of this, but this is going to the other extreme of super high integrity. So that they're seeing incredible things here that they couldn't possibly see any other way.
And this kind of technology has completely changed and transformed the understanding of space. The term dark matter really just means unknown. They don't understand the nature of it because it doesn't fit into the model that we have.
Well,
But
the model,
the whole model is in the midst of this huge transformation because of this technology allowing greater sight, than
yeah. What Ron Cowan did was he showed the dark matter. I showed you an electron that had 10 strings. The dark matter has 5 strings. So it doesn't vibrate as much, and it's in this orthogonal 10-dimensional subspace to the subspace that we're in. So the matter subspace is orthogonal perpendicular to the dark matter subspace, which is what Ron showed. And he was able to observe these things. And the other little quirk about dark matter is there's no charge. So dark matter just consists of the things that are called subquarks. You put 3 of them together to get 1 quark, and 3 quarks make in the world of matter, they would make an electron or a neutron or a proton, depending on the type of quark. But in the world of dark matter, they turn out to be they look like ferns, like flowery ferns, except they can be hundreds of thousands of kilometers long. Ron observed these in deep space. These things are running around there.
Seaweed frogs.
So
growing.
Yeah, they've got to accept what Ron did. They don't buy into it yet. They're still in denial. So anyway, so that was that.
The technology's catching up.
Right.
with the East, really.
Well, it's still got a ways to go. So now here, let's see, hold on. I'm going to go back to our class. And here we have, let me do the share screen. Okay, here we had, this is what we started with, and we did some of this. This is a, let me just go into, get this thing out of the way.
Are you recording?
Oh yeah, we're recording.
Sharing.
Yeah, so here, what we're talking about is modern biology. And this is Darwin who's here. And what happened out of Darwin was there was a big push to look at the piece parts of biology, which turned out to be DNA and RNA. And so here's a picture of a so-called double helix, the two DNA strands. And to put this in perspective, you have 50 trillion cells in your body, and each cell has one of these things in it, but they're 6 feet long. But when they fold up, they're under 1 millimeter. So the thing that's hidden here in the actual encoding of the DNA and RNA is the folding. How do you fold this thing together to go from 6 feet to under 1 millimeter and have it fit and so forth? So there's something amazing going on here. And so this is the direction that modern biology took. But
And then completely different things end up next to each other. You know, they could be the whole, 6 or 7 feet apart, or they could be right on top of each other once they're folded up.
So what we covered was, here's the mainstream path of what is modern biology, genetics, genomics, et cetera, and incredible engineering that goes with this. But there's a totally separate path that we think is really more fundamental and will come out into play in times to come, especially in terms of medicine and actually curing people of things. And this alternate path, Marjorie wrote a very nice essay on Darcy Wentworth Thompson. Do you want to talk about him?
Well, this was around 20 years ago. This is the kind of thing that Bart and I had in common, which at first we wanted to do this class as a debate, but we realized we agree too much. We can't debate. We would just end up saying, yeah, really. And then this, so we were just always destroying the debate. But we had both read him. Bart read him in graduate school, and I was asked to do an essay for an academic publishing company in 2002. And I was just in love. It was, I guess one of the juiciest things I had ever come across. And he was totally enamored of the Fibonacci sequence and aware of that in nature and space. It's a slightly different spiral or ratio that seems to govern galaxies, but it's very similar, and solar systems. So it's some kind of ratio, if it's not that one, seems to always be at work wherever there's an additive cumulative growth process. It may not show right away, like nature looks really chaotic. And if you look at a plant, you think, well, there's no order there. The leaves are just, but the leaves grow in a spiral around the stem. And, and then if you look at it microscopically, and as a youth, he was really into these little fossils that are all along, he was Scottish, in all the Scottish Isles, these tiny little fossils kind of form sand along the shorelines. And they have these spirals in them in different expressions in different ways. And he had this way, can we show any visuals? I think a picture is worth 1,000 words.
Just we could show you a conch cell, for example, just but I think you've seen a kind. So I just wanted to pick up on what Marjorie's saying. This is not it's a new idea for biology, but this is really, I think, due to someone like Plato,
Right.
who talked about the Platonic forms. He said these forms were everywhere. So in 2 dimensions, you have the equilateral triangle, you have the circle, you have the square, you have the pentagon, you have the hexagon, the dodecahedron and the duodecahedron.
Those are
in
Darcy's favorites.
in a plane, and then in 3 dimensions you have a pyramid with equilateral triangles. You have a cube. You have the octagon forms this 3D thing and so forth. And these things get repeated again and again. Nature just reuses them. Okay, was what was going on. That's what Darcy Wentworth Thompson picked up on was that the how can you you end up with a head. Okay, the head is the head is the head. Well, we're in I just showed you the strand of DNA. Show me where the head is. Okay, he's saying that there's some overarching form that's guiding all of this, that's driving it. And that led to these other people, but in particular, this guy, Robert Becker, here did a lot of work with the electric field of the body. And he said, this is really controlling a lot of stuff. Now, you could read all the papers you want to about Watson and Crick and DNA, and you won't find the word bioelectricity anywhere. It's not there. So anyway, so this led to a lot of other people looking at this. And the latest one is Michael Levin, who's at Thompson University. He was born in Moscow. So he got pushed out in '89 with his parents because, of course, he's Jewish. But they were able to get out. And what he's doing, I think, is picking up on all these ideas 100 years after Darcy Wentworth Thompson, but he's got the advantage of computers and the advantage of artificial intelligence and the advantage of computer graphics, that they just didn't have. All of the real donkey work he's taken advantage of, beaucoup. So that's who we're going to go look at here is Michael Levin, and I'm going to go cut over to a video for him right now if I can, and he'll talk. Let's see, is this it? Yeah, this is one I just noticed he did 3 days ago. Okay, 3 days ago. Let me see.
You've been in a little contact with him, haven't you?
5 days ago, and it's already got 3,000 views. Okay, so let me just tee this one up, and you'll start to see.
He was given
He's really very much
launch.
Yeah,
to do whatever he.
Yeah. Paul Allen gave him a
really
check for $10 million and said, I like what you're doing. I don't want to have you waste time writing up proposals for money.
And
So
he
just
doesn't
come
have
to
to
me
get
and
involved
I'll
with
find you.
politics.
Okay.
There's a lot of politics in science.
Yeah. So
And it can be extremely dogmatic. I mean, just as much as any, religion. And it's not based on science. It's not based on truth. But they act as if it is. And a lot of it is money based. A lot of it is about the politics of the grants involved and things like that. So that's why he was given this amazing opportunity to just do pure science.
So here he's going to be talking about this goes almost into philosophy, but I just want to show you where he's coming from. I'm going to put the captions
So Mike
on.
Levin, Vannevar Bush, Distinguished Professor of Biology at Tufts University, an Associate Faculty at Harvard's Weiss Institute, and the Director of the Allen Discovery Center at Tufts.
Thank you so much. So I'm going to try to talk about some ideas today in fairly brief format given the time constraint. So if anybody is interested in the data, the software, the videos, everything is at this site here. And then this is my own personal blog as to what I think all of these things mean. So one of the things that we are interested in is augmenting this idea of the search for extraterrestrial intelligence with what I call SUTI, which is the search for unconventional terrestrial intelligence. I'm going to tell you that I think that there are minds all around us, which we are currently very bad at recognizing. And there are two key things. One is to have the right perspective, and I will try to widen our perspective today. This is a pile of forks and knives and spoons that, if you have the right perspective, you actually see a pattern that is not at all obvious from first glance. And the other issue is that I'm going to claim that any kind of a cognitive assessment of a system, whether we think it's what degree of intelligence it has, or cognition, or whatever, is really an IQ test for us too. It's a bidirectional interaction that requires the observer, namely us, but also parasites, constant physics, and so on, to have the ability to detect the agency of other systems. So if we don't see it, it may be the problem may be with us. And so what I would like to do is to contribute to a kind of unification. So this is the electromagnetic spectrum, and prior to having a proper theory of electromagnetism, we thought that there were many diverse phenomena. There was light and magnets and static electricity and lightning. We thought all of these were different things, and there were huge areas of that spectrum to which we were completely blind because our own evolutionary history does not allow us default access to it. And then what the theory of electromagnetism allowed us to do is to, first of all, unify all those phenomena and realize that even though they seem different and they look like they belong in different categories, they're actually all the same thing. And the different aspects of the same thing, and it enabled technology that lets us now access other parts of that spectrum, so go beyond our own limitations and do things that improve the quality of life. And I think this is a very good metaphor for where we currently stand in our ability to recognize things that we're interested in the cognitive sphere when they come in unfamiliar embodiments. And I think this is a kind of mind blindness, and what I think is important is to develop conceptual frameworks that help us get around it, and the technology that helps us to engage with that in a very practical way. So what I try to do is, among many other things, is to develop a framework that allows us to think about very diverse kinds of minds all at the same time. So yes, apes and the birds and things that we're familiar with, and maybe an octopus or a whale, but also some very strange creatures, colonial organisms and swarms, and then, very importantly, engineered new life forms, so synthetic biology, also potential AIs, whether software or robotic, someday maybe exobiological agents, and then something else that's quite interesting that I probably won't have time to get to today, which is erasing the distinction between patterns and objects. And so there are many interesting patterns that I actually think have agency that are very hard for us to recognize. So I'm certainly not the first person to try to do this. Here's Wiener, Rosenbluth, and Bigelow back in 1943 trying for a scale. Now they were working with certain phase transitions, they thought, but basically a cybernetic scale that goes all the way from passive matter all the way up to second order metacognitive humans and whatever is beyond that. So what I would like to do is to develop this kind of framework with the idea that, first of all, it is not just philosophy. I think philosophy is very important, but it is not only philosophy. It is good to the extent that that framework will move progress forward. In other words, I want to see it drive new science, new engineering, and better ethics. In other words, it has to be practical. These things have to be they have to demonstrate their utility at the bench. They can't just be ideas. And the details of much of it, at least the first version, is all here. So what I'm interested in is a view of the spectrum of cognition that I call the axis of persuadability. And I say persuadability because I would like to cash out cognitive claims as interaction protocols. In other words, when you tell me that a system has a certain degree or kind of cognitive capacity, what I hear is you are announcing what is the interaction protocol that you will have with it. You will use tools of physical rewiring, or perhaps cybernetics and control theory, or perhaps behavior science, or perhaps psychoanalysis and friendship and love and whatever else is up here. So what you're saying is I'm going to make a hypothesis, I'm going to use a particular set of tools, and then we are all going to find out how that went for us, for both sides, which means that this kind of thing is to be decided empirically, not by trying to hew to ancient philosophical categories or ideas about what kinds of substrates have some sort of a monopoly, or substrates and origin stories such as evolution would have a monopoly on making minds. We can't decide this out of an armchair. We have to do experiments, and this is what we've been trying to develop. Another way of looking at this actually is what happened to the notion of numbers. So people started with counting numbers, and then somebody came up with zero, and then eventually negative integers, and eventually fractions, and then irrationals, and so on. And what happens every time you do this is several things. First of all, you have to make a conceptual leap. You have to break the old category that demarcated the kinds of things that you thought you were dealing with. And it's actually quite painful. I mean, people died over irrational numbers and things like this. All of these are disruptive. And so what I would like to talk about, and I'm only going to get to a portion of this today, is learning to recognize minds in increasingly more unfamiliar embodiments that are weirder and weirder and more unconventional. But I think that's exactly the journey that we've been on in science and in mathematics. And so, I'll probably only get to here, but let's do some of this. The one thing that actually frames a lot of these kinds of discussions is this ancient pre-scientific vision, which is this is called Adam names the animals in the Garden of Eden. And the idea here is very simple. There are discrete natural kinds. We all know what these animals are. And they're all brainy sorts of things. And Adam, of course, is different from them. But the most interesting part for me is that it was his, in this biblical story, it was his job to name the animals. God couldn't do it, the angels couldn't do it, Adam had to do it. And that was because, first of all, in those ancient traditions, naming something means that you've discovered its true inner nature. Right? That's the deep meaning of naming something. And also, because Adam was the one that was going to be living with them. So I think that's the part of this that's completely wrong is these very a focus on these discrete brainy kinds. But what's I think deeply right about this is that we are all going to have to name, in this sense, a very wide and very strange set of beings. So first of all, let's just start with brains. Pretty easy. We see something like this. So this one, this little creature is going to set up a little accident scene here, like this. He puts the thing on his neck. In a minute, he's going to check to see if his parents are watching. He has a very good theory of mind. He understands what the reaction is going to be, and so this kind of stuff is obvious. Medium-sized objects moving at medium speeds. They're similar to us. It's pretty easy for us to detect minds in animals that do this kind of stuff. Actually, even that story is not very simple. There are numerous cases which we reviewed, Karina Kaufman and I reviewed recently, where the mapping between the brain and cognitive performance is completely broken. So there are patients with very little brain tissue like this, and actually normal or above normal cognitive performance. So even in brainy animals, the story is not at all simple as to what's going on. And this is a theme that's going to come up towards the end, is that in certain cases, you actually get a lot more performance than the hardware seems to warrant. And that raises some interesting questions about why the current models of neuroscience don't predict this. It's not that this can't be hammered in like epicycles into the current model, but the model certainly doesn't predict that anything like this is going to happen. So there's a problem. And this is the problem, that you often get more performance than you can account for with its history or with the hardware. Let's leave that aside for a second. And what we're going to talk about is a model system that we've been studying, which is cells as a collective intelligence living in anatomical morphospace. It's a kind of an intermediate stepping stone to some of the much stranger kinds of things that I want to talk about, because to some extent it is similar to us, and in many ways it's very different. So we're going to look at this as a model system. First of all, let's remember that we are all composed of an agential material. In other words, at many scales, we are not made of passive matter. We are made of components that have their own agendas. So this is a single cell. This is a single cell. It's called a lacrimaria, which is a free living organism, but you get the idea. Our cells, like this one, there's no brain, there's no nervous system, but there's extreme competence in its tiny little agendas. It has a small cognitive lexicon of time and space that it cares about, and it's very good at handling things in that scenario. Now, it turns out that even within the cell, that thing already is made of a material that has some competencies itself. So just molecular networks, never mind the cell, never mind nuclei or synapses or any of that stuff, just small molecular networks by themselves can do 6 different kinds of learning, including Pavlovian conditioning. They can do habituation, sensitization, associative learning, and so on. In fact, if you train them, you will find out that that training increases the causal emergence. So phi-D, for example, will increase over the network. So actually, training within an environment and forming associative memories actually reifies the nature as the nature of this system as something distinct from the sum of its parts, as quantified by recent advances in causal information theory. So even the material of which the cells are made themselves already has certain cognitive capacities. We in the lab are taking advantage of this to train the networks and to do things like drug conditioning and so on. It's a very practical kind of thing. This is not just theory. It's very it has many implications for biomedicine and so on. And you can see all of that here. So what we have actually, and I could spend hours talking about each level, but fundamentally we have this thing that I call a multi-scale competency architecture, that basically all the way from the molecular networks to large groups of organisms, at each level what you have are problem-solving agents that have specific ability to pursue goals with various degrees of ingenuity. Each of these levels solves problems in different spaces. However, they don't all live in 3-dimensional space the way that conventional behavior does. They live in some other spaces. For example, the space of gene, they navigate using the familiar loop of sensing, decisions, making decisions, and then taking actions. They deploy that loop in transcriptional space. So high dimensional space of gene expression, anatomical morphospace, the space of possible three-dimensional organizations, physiological state spaces, metabolic state spaces, and so on. Biology does this in many spaces, and it was doing this long before nerve and muscle came on the scene, by which conventional behavior that we're all pretty obsessed with the three-dimensional world, and we think embodiment, especially for AIs. If it doesn't have a robot that's bumping around the 3D world, we say, well, that thing's not embodied. I think this is a fundamental mistake, and it's true also of organoids and things like this. Just because it's not rolling around our three-dimensional world, and in fact, it may be navigating these other spaces that are very difficult for us to visualize. But we're making tools to help us do that. So let's just talk about this anatomical morphospace for a little bit. The hypothesis here is that basically, I used to have my students do this by hand as an exercise, and then we made a tool that you can play with here called Field Shift. Basically, you can take almost any neuroscience paper. In other words, in the field of neuroscience, where we study the sort of uncontroversial example of cognition, meaning brainy humans and animals, it turns out that all of those concepts are actually very well mapped onto some very different problem spaces. So if you take a neuroscience paper and you basically change all the scales from milliseconds to minutes, and you basically, instead of every time it says neuron, you just say cell, you actually get yourself a very nice developmental biology paper. Because the same kinds of things that are happening in your brain to underlie your cognition, meaning the cognitive glue that electrophysiology enables, those same exact processes were functioning, because they're ancient evolutionary precursors to brains, were functioning to act as the cognitive glue to cellular swarms in morphogenesis. That is where our brain gets all these amazing tricks. So I just wanted to show you a couple of examples. Again, we could spend hours on some of these amazing things, but just very simply, just to remember that embryonic development. So you start here as an egg, and then you get to this. It's very reliable. But that is not why I'm going to argue this phenomenon involves the behavior of a collective intelligence. It's not because it's reliable. It's not because there's emergent complexity that increases here. Those are cheap and easy. It's very easy to get those. What I'm talking about specifically is problem solving in terms of intelligent navigation of anatomical morphospace. One thing you see immediately is that if you cut this thing in half, it does not do what context insensitive mechanisms do, which is the same hardwired thing. No, actually, if you cut it into pieces, you don't get half bodies. You get perfectly normal monozygotic twins, triplets, because all of these things can navigate the morphospace to this ensemble of goal states that we call a normal human target morphology from different starting positions, avoiding various local maxima. That process is actually much more general. I think development is actually just a special case of regeneration. It's extreme regeneration, where you have to repair the entire body from just one cell. And again, navigating anatomical space, you start at where a single cell is, and then you end up at the space of the configuration of whatever kind of creature you are. But some creatures do this in adulthood. So again, this axolotl, if you amputate the limb anywhere, the cells immediately can tell that they've been deviated from their correct position. They act really quickly to reduce the error, and then they stop. And this is the most amazing thing about regeneration, is that it knows when to stop. When does it stop? It stops when the correct salamander limb has been completed, meaning they stop when they are back at the same position in that morphospace, meaning this is a homeostatic process. It's a process of error reduction, and it's a process of pursuing a specific goal. Now, who has this goal? The individual cells do not. No individual cell knows what a finger is or how many fingers you're supposed to have, but the collective absolutely knows, in the only sense that matters, which is the practical question. If we try to deviate it from its goal, is it going to try to get back? And if so, with what degree of ingenuity? That is the question, and I'm using William James's definition of intelligence here, which is the ability to pursue the same goal by different means, and some degree of that ability. In fact, it's important to note this is in no way about simply repairing damage. This is something much more interesting, because if you take, for example, a tail and you graft it onto the flank of this animal, it remodels itself into a limb. Okay, and the most interesting part of this is that the cells at the end of the tail here, they become fingers. Now, locally, there's nothing wrong with these cells. They're tail tip cells sitting at the tip of a tail. Why do they change? Why do they become their fingers? Why do they become fingers? And that is because this whole system, again, at the scale of the whole animal, is an error minimizing, goal seeking system that tries to maintain a specific state in anatomical space. And in order to do that, it does something that all good cognitive systems do, which is that it harnesses molecular mechanisms towards these high level goals. So when you and I wake up in the morning and we have very abstract scientific, social, financial goals, whatever, ultimately all of those, in order for us to get up and walk and execute on those goals, they have to end up changing the chemistry of our muscle cells so that the ions flow across the membrane and we can get up and walk. Voluntary motion is an example of where high level goals that belong to the collective, not to the individual cells, are able to harness the biochemistry in towards those goals. The exact same thing is happening here. What the system here has ascertained is that what goes at this position is not a tail, it's a limb. And so what it does is it harnesses all of the downstream molecular biology needed to tell these cells to start turning on and off different genes, to start migrating around and so on, to create fingers. So top-down control of the molecular biology in line with abstract high-level goals that neither the molecular circuits understand nor the cells. It is a multi-scale control system where the high levels bend the option space for the lower levels, and then they do the thing they do, but the local order obeys the global plan. That's the whole point of this system. And so how does this work? Well, in the brain, we at least know the beginnings of how it works. We understand.
Doesn't that also imply some kind of holographic relationship to each part can become another part? That means it contains the whole in some way.
Yeah,
and
well, that's what happened
potential.
with Bruce and Ron. They were twins.
Right.
That's how it came about. So this is okay.
That there's hardware that looks like this. So
It's like
electrical activity across the muscle membrane sets up voltage gradients, which then do or do not
jump
across
in
these
a
electrical
lot,
synapses, and that underlies a bunch of electrophysiological dynamics in the brain that neuroscientists hope to read out in the project of neural decoding. And the idea is that these electrophysiological dynamics in some way hold and support all of the cognitive processes that need to go on when this creature navigates three-dimensional space. So what does this electrical network do? It sends signals to muscles to move you through three-dimensional space. Where did this amazing trick come from? Well, it turns out that evolution discovered this long before neurons and muscles appeared, and every cell in your body has ion channels. Most cells have electrical synapses to your neighbors, and all of this kind of stuff, the electrical communication that forms networks. Networks are very good at integrating information, storing memories, making decisions, context-sensitive behavior. This was all discovered by evolution around the time of bacterial biofilms. It is absolutely ancient. And what happened later, once nerve and muscle came on the scene, is that it got pivoted into 3D space instead of anatomical space, and it got sped up quite a bit so that animals could run around and try to eat each other. But basically, it's the same scheme where this electrical network gives signals not to muscles, but to all of the cells of your body during embryonic development, during regenerative repair, during cancer suppression, and then we'll talk about all those things, and tries to control the navigation of this group of cells through anatomical space. That's what it is thinking about. The electrical networks in brains, in animal brains, think about movement. The electrical networks in your body long before you had a brain thought about moving your configuration through anatomical space. And once when we wanted to pursue this almost 20 years ago now, we developed the first molecular tools to read and write the electrical processes in non-neural cells. So basically take the tools of neuroscience and apply them outside the body to see if we can do the same thing. And so what I'm showing you here is one example. These are voltage sensitive fluorescent dyes that allow us to, without having to poke every cell with an electrode, it allows us to catch all the electrical conversations that these cells are having. These are cells in the early frog embryo. These are explanted cells making some decisions about crawling off on their own versus joining this morphogenetic event. And so we can read these events. We've made all kinds of simulators so that we can have multi-scale simulation of what the information is that's being processed in these networks. We can connect them to ideas in dynamical systems theory and connectionist machine learning, such as pattern completion that you just saw in the regeneration example, and so on. But this is just to show you some examples of what that actually looks like. So here's an early frog embryo putting its face together. This is a time lapse. And if you take one frame from this video, you see very clearly, you can catch a memory of what the frog face is supposed to look like. So long before this tissue was regionalized by gene expression, you see here's where the eye is going to go, here are the placodes, here's where the mouth is going to be. And this voltage pattern controls the morphogenesis of the face. If I change this pattern, the face changes. I'm going to show you that momentarily. This is a snapshot of the kind of thing you would do in a human brain if you wanted to catch specific correlates of cognitive states that are remembering goals or sensing visual inputs and things like that. These things are not just the cognitive glue. Not just the cognitive glue within a single embryo. They are also the kind of thing that binds together groups of embryos. So you can see each one of these things is a separate embryo. You can see these waves propagating between them. It works on multiple levels, and in fact, groups of embryos have their own identity. They resist teratogens, for example, much better than individual embryos.
It's all like
we have some understanding now of how that works. The collective knows things that the individual animals don't know. And this is what I think neuroscience. I don't think it's about neurons at all. I think it's about cognitive glue and different policies and mechanisms that can be used to bind competent subunits into higher units. Now, tracking and measuring these things is all well and good, but you actually have to, the important thing, of course, then is to do functional perturbation experiments. So how do we change this? We don't use any applied fields. There are no waves, no magnets, no frequencies, nothing like that. What we do is we hack the interface that the cells are normally using to control each other. And that means the electrical synapses and the ion channels. And we have optogenetics and pharmacology and all the tools of neuroscience that we use in non-neural cells to hack this. And when we do, I'm going to show you a couple of specific examples. One quick thing I want to show you is the takeover of a cell by a particular very convincing pattern. So the color here is the voltage state. These are two frames, and I'm going to play the movie. So this cell is red, and what in the next frame you see that as soon as it touches the rest of these, it changes its voltage to be just like them. It only takes a tiny bit of just a single touch. So here, watch. So this thing has its own different voltage. It's going to come over here. This guy reaches out to touch it. Bang. That's it. Now it has changed. It's got the same bioelectric state as the rest, and now it joins into this thing. These messages are. It only takes a tiny bit for specific messages to radically change the electrical state. And this is the kind of thing we take advantage of when we take an early embryo and we use potassium, for example, potassium channel RNA to establish that little voltage spot that I showed you in the electric phase a few minutes ago. We will put that, for example, in cells that normally would be gut. And what it does is it tells those cells, no, instead of that, an eye goes here. And sure enough, they build the eye. They do all the downstream steps. They turn all the genes, all the stem cell stuff happens. We don't control any of that. We don't need to. Lens, retina, optic nerve, everything will be there because we gave it a high level prompt, just like I'm giving you now as I'm talking to you. I'm not worried about arranging your synaptic protein so that you can understand me. You will do that yourself. I'm using this thin language interface, and that's exactly what's happening here. We provide a top level signal that says you should be an eye. Not only do they make this eye, if there's not enough of them, there's only a few here, these blue cells are the ones we injected, they will actually recruit all their neighbors, just like other collective intelligences do. Ants and termites, they recruit their neighbors to solve a bigger task. These guys will do the same. We don't need to teach them to do that. They already do it. Okay, so the material, this material actually has many interesting competencies. And what we're learning to do with these bioelectrical stimuli is to talk to the material, literally to communicate messages that have meaning to the cells. There's nothing about that voltage spot that is essential to being an eye. It is a completely arbitrary symbol, and it only works because they interpret, the rest of the cells interpret the signal in a particular way, which we now are beginning to understand. I want to point out that what's happening here is actually quite interesting. The conventional molecular biology story would be a set of well-known events that lead from a voltage change to the transduction machinery to the identity of the cells. But that's actually the same thing as trying to describe a conversation between two people in at the level of physics of some air molecules moved around. It misses the whole point. What actually happened here, it's not false. I mean, those molecular processes did take place, but what's much more interesting than that is that at a higher level, what's happening here is actually a battle of goal states. And I'll point out this thing here. When we inject our let's say the eye-inducing signal, we inject it here. When you look at an early embryo, looking at the blue color here indicates which cells are turning on this gene called RIX1. It's a very early step of eye development. So who's on their way to a sort of a beginning eye development? What you see here is that you see here. Okay, there's going to be 1, 2, 3, 4, maybe 5. There's going to be all kinds of ectopic eyes. Fantastic. But if you wait, eventually there will be maybe only one or maybe none. And if you track it down, what actually is happening here is that, okay, these cells are saying to their neighbors, we should make an eye. We should all make an eye. But the rest of the neighbors, actually, as a part of a cancer suppression mechanism, they resist and they say, no, we should be gut or skin or whatever they are, because it is an ancient mechanism for cells to look at their neighbors, and if the neighbors have some kind of weird voltage, you assume it's an incipient tumor, and you try to normalize them by equalizing the voltage. So the cells resist. So there are two goal states here. There are two patterns. One is the eye pattern. One is the let's say skin or gut pattern. And what's actually happening here is a competition of those two patterns for the attention of the cells. Sometimes the eye wins. Sometimes the default wins. But you can watch this. You can watch these patterns as I just showed you in those two cells. In that case, the single cell was taken over. In some cases, it's not. And so we are now on the search for ways to manipulate those decisions, because that's the key to a lot of regenerative medicine, where you can get the cells to buy into your goals, not to resist, not to try to adjust elsewhere, as many of our drugs cause side effects, but to actually convince the cells that this new goal state is what they should be doing, because they do not simply take it up. They often resist. And it's very much, all of these are very much these kinds of cognitive processes. So what I've shown you up until now is a way to change the more. Again, I could show you many for hours, but I showed you one example to provide specific prompts to the network of cells to get them to build something that they already know how to build. For example, an eye. And I want to show you something completely different, which is this little creature. If you look at it, you might think that I got this out of from the bottom of a pond somewhere. But if you were to sequence it, you would find that it's 100% Homo sapiens. Okay, it is 100% normal human genome. These are cells that were taken from an adult human tracheal epithelium. They, when liberated from the rest of the body, they form these little things called Anthrobots. They don't look like any stage. They don't act like any stage of normal human development. They first of all, they have 9,000 genes expressed differently than the tissue that they came from. In other words, their transcriptome is radically different. About half the genome is now completely different. We did not change the genome. We did not put in any synthetic biology circuits. We did not put any reprogramming factors. There are no Yamanaka factors here, even though Anthrobots are younger than the cells they come from. We didn't do any of that. What they do is express new genes based on their lifestyle. Their lifestyle has 4 different behavioral patterns that you can draw epithograms of, and they can do many weird things. One of the things they can do is gather together into this superbot cluster and heal neural wounds. This is a big scratch that we made in a neural tissue. Here, if you lift them up, you can see what they're doing. They're healing this. Now, whoever thought that your tracheal cells, that sit there quietly dealing with the mucus and things like that, could form a self-motile little creature that can do stuff like this. So this is another place where this idea of getting more out than you put in comes from, because we know where you paid for the computation to design a human. It was in all of the evolutionary interactions going back, a billion years against the environment. But there was no selection to be a good Anthrobot. There haven't been any Anthrobots. When did we pay for the computation needed to have this gene expression, these behaviors, this kind of capacity, just beginning to be studied, their memory properties and so on. It's there's some very interesting issues here. And so the last thing I'm going to say is this. We're gonna take one step further, and we're going to talk about this issue of the plasticity of life and so-called machines. This is where I tend to lose a lot of people. A lot of audiences are with me up until now. Amazing plasticity of life and things that weren't directly selected for. Lots of people like that. Here's the next part. Because we know that this thing that we call this features very prominently in lots of discussions of philosophy of mind and so on. The human mind. Okay, this is at the center of a smooth and continuous gradient leading up from a single cell in evolution, of course, from a single cell in development. But there's also another gradient, which is that, again, through slow and gradual changes, both technological and biological, it becomes really pretty impossible. We already knew it was really hard here, but it becomes pretty impossible to say when this actually ends and something else that we want to call with a different name begins. This whole thing is not about machines and AI. It's about this kind of thing. It's about the inevitable symbiosis between human evolved and designed material, and this idea that we're going to try to be looking at these things and trying to classify them in these ancient categories that basically come from a time when nobody thought that this was possible. So we need to think hard about what is it that we actually mean when we're trying to given these continua. What is it that we actually mean when we try to put names to these things, and what do we want these names to do for us? And I don't have time to go into this in detail, but I recommend this paper if anybody's interested. Basically, what we found is, and I did this on purpose. I wanted to, for sort of maximum shock value, to take the simplest, dumbest example, most minimal example that we could, and I chose sorting algorithms. If any of you have done coding, this is like bubble sort, things like that. They've been known for, 60, 70 years. Every computer science student studies them. The important thing is that they're completely transparent. Unlike biology, where there's always some kind of hidden mechanism to be discovered, these things are like 6 lines of code. They're completely deterministic. You can see exactly what they're doing. There's no magic. And yet, what we found out is that if you look at them the right way, and this was my claim at the very first slide, you need to know how to look. If you look at them the right way, you see 2 things. You see the thing you force them to do, which is what's in the actual algorithm, which is to sort the numbers. And they do that. Now they have some competencies you didn't actually teach them. For example, delayed gratification. It's nowhere in the algorithm, but they do that. But that's delayed gratification. Navigating a space is an important cognitive property. So that's something. But what you'll notice is that they also do something else, and it's called clustering, which again is nowhere in the algorithm. Okay, they do this as a kind of the way that I think about this now is in terms of intrinsic and extrinsic motivation. In other words, there are the things you forced it to do with the algorithm because they do follow the algorithm. But at the same time, they do some other things. They have these weird side quests that are neither forbidden nor prescribed by the physics of the world that they're in. So I'm going to claim, and there's a lot more that could be said about this, that it doesn't take life, it doesn't take cells, it doesn't take huge complexity to have emergent capacities that we would recognize as elements of what cognitive science is supposed to study, including problem-solving competencies, and also a kind of intrinsic motivation, so that when people say these AIs, they only do what the algorithm tells them to do. They're not like us. We have freedom. They just do what the algorithm is saying. My point is, and even bubble sort does only what the algorithm says it should do. Okay, nothing only does what the algorithm says it should do. So we need to really rethink some of this stuff. And my point is just this. Just like Magritte was telling us that our representation of things is not the thing itself. This is our graphic artist. I had him draw this thing, which basically says this is not a Turing machine. Because my point is that our formal models of computation, so Turing machines, algorithms, those kinds of things, do not properly describe quote unquote machines any more than the laws of chemistry describe what our minds are capable of. It is exactly the same problem. We do not understand the capabilities of matter, and it's because we somehow assume that for living and conventional beings that the molecular facts don't tell the whole story. But when it comes to these simple things, we've somehow decided that those formal models are good enough to tell you everything you need to know, and they're not. In no case are they good enough. And so I think that, and so I'm just going to stop here and say that I think the future Garden of Eden is going to be a very weird and very different place with all kinds of beings in radically different embodiments. And just as a last kind of a topic for provocation, I will just say this: that in biology we love two sources of information, because you might want to ask, where do the behavioral and anatomical patterns come from? The typical biological answer is two things: physics. So physics requires certain things, and a history of selection. There's a long history that tells you why this creature is exactly the way it is. And I just want to remind us all that there is a third. There's a third option. There are facts of mathematics. For example, this particular shape that is encoded. This is a Halley plot, that it comes from this little tiny seed. In complex numbers, this is what that looks like. This has a very particular pattern. If you wanted to explain why that pattern, you could not use any fact of physics to explain it. You could not use any fact of history to explain it. There are mathematical structures that have patterns that are not explained by anything in the physical world. Some of them are very organic, so that's kind of fun. But the bottom line is that it is not just about physics and selection. I don't think the physical world is closed, and this has a precedent. There's a lot of mathematicians. So Plato's mathematicians.
Excuse me, Michael, you have 10 minutes. 10 minutes left.
Okay, I'm almost done. I'm almost done. One couple more slides and I'm done. So there is a precedent for this, which is this idea that mathematicians are not just inventing, but actually discovering a space of structures. And my point is about all of this stuff is that the reason that in biology, but also in even simple things like sorting algorithms, the reason you get more out than you seem to have put in is very simple. Because all of the things we build, whether they be machines, embryos, biobots, whatever, AIs, whatever it is, what we're actually making are interfaces or pointers into a pre-existing space of patterns. And this is not just a random grab bag of emergent surprises that we can catalog in our big book of emergence, but actually a structured space that can be investigated the way that mathematicians investigate their region of that space. We can use these biobots to investigate regions of that space that ingress into our physical world. And so in the future, because of the plasticity of biology, pretty much any combination of evolved material, engineered material, software, and these patterns, which do not come from the same place as these others, will make some kind of embodied mind. And everything we're used to is a tiny little corner of this option space, and we better get significantly more sophisticated in being able to relate ethically and productively with beings who are nowhere on the tree of life with us, and have radically different embodiments and views. And so I'll stop here and just say this. I think that the sharp categories that people like around life, machine, and so on have, as a matter of empirical fact, have not enabled, they've constrained new discovery. Okay, I've yet to understand what any of these distinctions have helped us discover, but dissolving these boundaries by applying tools across disciplines have now led to many practical advances. I think that intelligence in the broad sense is pretty much everywhere. Again, we can't just say it's there. We have to do experiments, perturbational experiments. But learning to detect it is actually essential for progress in biomedicine and ethics. And so we now have a research agenda that is paying off nicely. And the last thing I want to say is this: putting living things and machines on one spectrum does not mean that I'm equating life with the kind of simple machine metaphors that people like to use. My claim is the exact opposite. I say that those kinds of metaphors, so the basic computationalist metaphors, are inadequate to deal with what we mean by life and mind, but they are equally inadequate to understand even simple machines. These ingressions from the space occur very early on, and basically these we have to understand that our formal models are not good even at the very low end of that spectrum. So I'm not trying to mechanize biology, quite the opposite. I'm trying to point out that the special and semi-magical things that we see about life and mind actually extend pretty much all the way down. And so I'll just end here. If anybody's interested, there are lots of papers that go into this stuff in detail. And I want to thank the postdocs and the students who did the work. Lots of amazing collaborators that work with us. I have to do 3 disclosures. These are companies that have spun out of the work of our lab. So some of these philosophical ideas give rise to very practical, practical applications in medicine, environment, and so on. And most of all, I thank the model systems that we work with, because they do all the heavy lifting for us. So I'll stop there. Thank you.
So thank you, Michael, for this fascinating talk. We have actually only 4 minutes left. Okay, Josh.
First of all, really enjoy your work. Been following it for a little bit. I'd love to have where you started and where you ended, but as you already sort of prefaced, it's the interesting sort of middle part that I have a question about. Specifically, well, local field potential, for example. I understand that your work, as you clearly stated, doesn't apply any fields, but you wouldn't, I suppose you wouldn't necessarily, or would you deny that local field potential as a shared phenomenon amongst cells, whether neural or not, that it could not be at this higher order causal aspect of your model? And then furthermore, to one of your final conclusions about machinic approaches, wouldn't something like local field potential not be available to Turing equivalent state-based computation? Thank you.
Thank you so much. First of all, yeah, absolutely. Local field potentials are important. I love, for example, Earl Miller's work looking at those kinds of things in brain cognition. I mean, great. All of that is great. I agree with you that the standard Turing paradigm is insufficient for continuous field-based type of phenomena. But I will also point out that you don't need to get that exotic to find out that it is insufficient. We can already see it is insufficient when you pull together a 6-line algorithm for sorting numbers. You already find out that even there, our understanding of algorithms and what they do and the way they run simple machines, already there's something going on that is not captured by that. So yeah, I'm not at all arguing that the Turing paradigm captures any of this. It captures some useful things for sure, but like all models, it hides some very important stuff, and we are now in a position to investigate it.
Okay, so we have time for one more question. Timothy Eastman.
Yes, greetings, and thank you for a great presentation. I understand that wet chemistry is often just simply presumed to be mappable to classical physics framework and understanding without remainder, and yet clearly that's simply an approximation, and all systems at multiple levels are really quantum systems. We live in a quantum world. So some of these effects you're describing may really be, so to speak, tapping into fundamental quantum process or a quantum substratum. Any comments?
Yeah, they might, and I'm sure. And by the way, I will preface this by saying I'm no expert on quantum anything, but I will just point out the following. I'm sure that there are quantum phenomena that biology, in particular, makes use of. Perhaps some of the abiotic phenomena does well, but it does as well. But I will just say this: the kind of lack of closure of the physical world, meaning the falsity of physicalism, was already true in Newton's boring deterministic classical universe. We already had this. You don't need quantum, indeterminacy and all this kind. I'm sure it adds some cool bells and whistles, but already in the classical world, and I mean, Pythagoras and Plato already saw this. They understood very clearly, long before we knew anything about the cool, quantum physics, that there are facts that matter for the physical world that do not themselves originate in the physical world. They cannot be changed by anything you do in the physical world. There are mathematical structures that already ingress even into Newton's boring classical universe. So maybe there's new stuff happening with quantum mechanics, quite possibly, but physicalism was lost long before we got there, I think.
It was always too reductive. It
Thank
was
you.
always in
Thank you so much.
denial.
Thank you,
or
everybody. Yeah, great questions, and thank you for being here.
the X factor.
Yeah, if anybody has more questions, please email me, michael.levin@tufts.edu. I'm happy to talk about this stuff as much as you want.
Thanks so much, Mike.
Yes. Okay, so we have to move to the next.
So there you have it, Bruce.
You survived another session.
Yeah, he's an amazing guy, and he has a whole pile of graduate students, so he can really percolate a lot of ideas, and we'll see what happens. But I think he's going to deserve a Nobel Prize in biology over the dead bodies of a lot of other biologists. is what's going to happen. He's really getting into this, and we'll do another one of his, because there's so many of these things that he's got that are so I just think lucid. The example he took of the bubble sort with 6 lines of code, I'm quite familiar with, and it actually was developed at Bell Labs way back in the 50s. So it just shows you the power of these really simple ideas. How could 6 lines of code have anything? I mean, who's kidding who? What's going on? And yet you end up with this behavior. Or he talked about Newton with his the reality of what Newton did was you can only solve explicitly a two-body problem in with Newton's gravitational theories. If you have more than two bodies, you end up with these incredible complexities that people they lead to you developing these forms or templates to try and explain what in the world is going on. And Newton knew there was something more to this, but he was smart enough not to go there. All these other people went there and they discovered, holy cow, look what Newton missed. And it just shows Newton had good taste, that he wasn't going to make a fool out of himself by dealing with three bodies, four bodies, whatever it is,
But
and
that
so
is
forth.
the
And it's still,
situation.
in 1900 there was a contest: is the solar system stable? Yes or no? And Poincaré won the prize, but even he couldn't say, was it stable, yes or no? He just said, well, it might be stable for a couple 100 billion years, but it might collapse then. Okay, that was the best he could do. So go figure. Anyway, let's call it a night, and we'll try some more of this. But I think these notion of these archetypes that Jung talked about, or the forms that Plato talked about, or these templates, these ideas that Michael Levin is dealing with, I think are really fascinating and really get to the crux of, if we're ever going to do things like have people cure themselves, this is the direction we're going to have to go in, to energize your body to say, wake up and follow your correct template, and then the cancer will go away, or you'll live much longer, whatever it is. But we're not there today, obviously, but it's
Well, I think some people are.
it suggests that there's this is a much better path than just sitting there dealing with the drug companies, where the answer is always another drug, no matter what the question. So anyway,
What I wanted to say before we part company, if we can just have a little discussion, and I'd like to hear what Bruce would like to say. For me, this brings out so much of my sense of awe of the mystery of it all. That there is some order there, obviously, that's always been there. It's nothing new. And it's governing the way things have always been, and the way they're changing, the way they are, the way they stay the same, and the way they change and evolve is so much more mysterious than we would.
oh, this is just from Eric.
What did it say?
He thought we were going to deal with Jung tonight, and we did, but we dealt with it in a modern view.
Okay. Well, we could spend the rest of the
We have other stuff on Jung. I just wanted to go through, since Marjorie did such a nice job on Darcy Wentworth Thompson. I wanted to just kind of flesh this out some more, and then we're going to go back to Jung, and we'll deal with that. So, and there's a lot more stuff on Jung since we last covered it. It's just amazing what is on YouTube. These people just keep cranking these things out.
more is being published and written about by more people,
Yeah.
so there's more understanding.
Yeah.
But just as in this field, they're not really different fields. And as I love what he said about, no, this is being discovered as what's there. It's not what's being thought up. It's not an invention or a creation. It's a discovery. And I thought that was a really important point. And the more we look, the more mysterious it is, the more miraculous and complex and beyond comprehension. How could you ever understand it? What I just want to say, what the hell is going on? It's so mysterious. It's so complex. We have no idea how a salamander grows a new egg, how those cells organize themselves. And that's like, a microcosm. You know, I think
But, yeah.
humanity is acting like cancer cells.
Well, no, but what he pointed out was that you can just using these electric fields, you can stimulate this sort of growth to cure the cancer cells.
Right.
You know, so it isn't that he knows what he's doing. He just says, I'm just going to use the tools that are already there.
Well, there's some evidence that like just, you know, water drops in certain species can cause conception. You know, it doesn't even need to be a
Yeah,
sperm cell from another creature.
yeah.
Just the stimulus of the water on the ovum, you know, starts the division process.
Let's let Bruce chime in. How did your week go? Was it a good week? Oh, you're muted. Wait, Bruce, take the mute off. You're muted.
Oh yeah. Sorry. Yeah, oh, it was awesome. Like Mahamudra is a realization of a deeper level of your own mind that you already have, but it's very difficult to find or to get there from a meditation point of view. I found it, but I did it spontaneously. It wasn't through meditation quite a few years ago. But I want to talk about some in regard to your show. I knew somebody who cured himself of cancer, and he was talking to my teacher about it. So it was part of his own mind that did it, but his conscious mind was confused about it. And then
It was, I would argue it might be his soul that had the template in mind. And as you pointed out, his conscious mind was confused, but the template was there. And once he realized that, then everything kicked in, but he had to realize that.
yeah, yeah, but he did it. But then he was confused about it. And then when he was talking to my teacher, apparently they had a telepathic conversation. And then he was confused about that. So, but basically, another part of his reality decided to cure himself of cancer. It took about 2 or 3 years to do it, and it was gone. All the doctors were surprised. And all that
So the cancer had metastasized through his whole body. He was basically on his deathbed.
No, no.
And after 2 years, there was nothing.
He didn't say that. Bruce
I
didn't
can't
say
remember that well. He had it manifesting as a, it was physically manifesting on part of his body. And the whole thing disappeared. And he knew that his mind did it.
Right. But some part of his mind that wasn't conscious.
Was he meditating every day?
No, he wasn't that way at all. He wasn't even a student. But we happened to meet, and he told me about it. So I introduced him to Rinpoche, my teacher. This is many years ago, and they talked about it. But basically, he cured himself of cancer.
Did Ramchal Rinpoche have other people he met who did this? Probably.
Say it again.
Did your teacher Namchal Rinpoche, did
Yeah,
he
yeah.
have other people he met that had similar experiences that were able to, because he met many people.
Yeah. No, this is the only the one story that I know about.
Okay.
Well, I don't think it's far-fetched. I think it makes sense. And I think the part about it not even being conscious makes sense too.
Well, the doctor thought he's going to die
that
from it.
kind of thing.
Yeah, sure. Oh, well, that was why everyone points to the salamander as an example, and you cut off a foot and it regrows the foot. Well, how in the world does it do that? You know, if I cut my foot off, it doesn't regrow. Maybe it should.
Well, maybe there's a way. Maybe there's a way to tap into that.
Yeah.
So I don't know. I don't know why salamanders, but they you know, and I'm sure there's other ones if we looked around, but you know, anyway.
I
So
think we have so much more capability than we understand or believe that
Yeah.
it
Oh, there's
just
many
has
dimensions
to
that
be
nobody knows anything
developed.
about at all.
yeah.
Right.
So you
Well,
guys going to be on next week again?
yeah, we'll do another. He's got a bunch of videos. I just saw this one is brand new, but it was a nice little short summary, and he's got a longer one. So we'll run through that, and then we'll stop there and go back and do some Jung. There's a lot of new Jung videos out there too
Oh,
that are really amazing.
Excuse me, excuse me.
So just, you know, the people put a huge amount of time in, you know, thinking these things through, and the exposition is really excellent. So why reinvent the wheel, you know? Anyway,
For me, it's a wonderful sense of fun.
yeah,
But this is all going on.
Yeah.
It's just kind of
Yeah.
yeah. The whole thing. And that somehow this is apparently actually real and not just a projection. Maybe it is. Maybe
Yeah.
we're imagining it, but
Yeah.
it's still pretty amazing. It's
Yeah.
an amazing show.
Yep.
Whatever it is. And
Okay.
we're all having our own experiences simultaneously.
Well, we'll let you go. Have a good.
alrighty.
Welcome
And-
back to so-called
Yeah.
reality.
Yeah.
Okay.
Another week.
All right.
Good.